Hello and welcome to Agri-History. Today we'll talk about peaches. The fruit species Prunus persica, also known as peach, is a unique fruit tree species with edible and ornamental land races. In this video, we will cover the definitions of polymorphisms and phenotypes, and then talk about the domestication of peaches and why the species has the traits it does. A polymorphism is a state where a gene within a species genome has multiple forms, with each form of the same gene creating a different trait. Next, let's define phenotypes. A phenotype is the sum of a life form's physical traits. This is formed from both genes and environmental influences. Next, we must define isozyme analysis. Isozyme analysis is a technique used to determine what species a cell culture came from, using banding patterns made by enzymes as markers for what species it is. A process of elimination to determine the species within a cell line. This technique can also be used to determine if the cell line is contaminated with other species. The peach came into existence as long ago as 3000 BC. Based on current archaeological evidence. The cultivated peach is related to 5 wild species. These species are named Prunus myra, Prunus davidiana, Prunus cansuensis, and Prunus ferganensis. All these species, with the exception of Prunus ferganensis, have poor flavor. These species, however, can be used as a source for disease resistance or have use as rootstocks. Over the thousands of years of domestication, many cultivars have been bred. In fact, there's over 1,000 peach cultivars currently in existence. Many changes in phenotype have occurred in peach. One example is fruit size. There have been several examples of novel traits within the peach germplasm. Examples include flat fruit and non-fuzzy fruit. There have also been differences in flavor, and there have also been differences in flowers. For example, double flower type cultivars and cultivars with colorful anthers. These traits appear in peach, but not in the surrounding species, such as apricots, plums, apples, and grapes. This is despite the fact that the peach species has lower levels of genetic variation compared to other prunus crops. This is due to the capacity for peaches to self-pollinate, in contrast to the inability of most prunus species to self-pollinate, and the multiple genetic bottlenecks that have happened in recent breeding history has reduced the total genetic diversity. Using the current genetic technology, it has been discovered that the edible and ornamental forms of peach have fewer polymorphisms than the wild population. This seems to show that there was a genetic bottleneck when the cultivated population was separated from the wild population. The same data suggests that there's little to no apparent bottleneck once the peach land races were separated using the same genetic tools. It was discovered that the ornamental peach groups were derived from an already domesticated population of peaches. Using the genetic data discovered, it was revealed that the population used to breed all the eating type land races was also used in the breeding of ornamental peaches. Using ancient peach pits found in the Hemunu site ruins of China, it was discovered that at least the wild form of peach was eaten around 7,000 years ago. The earliest record on the history of peaches was in the Book of Songs, written 7,000 years ago in China. The first ornamental peaches were recorded 2,000 years ago. Using this archaeological evidence, the first domestication event of peaches happened sometime between 4,000 and 7,000 years ago, and the ornamental strain was bred 2,000 years ago. Now, let's talk about the peach's closest living relatives. Prunus myra is considered the oldest progenitor species of the cultivated peach, but the direct ancestor to the peach is unclear, although Prunus cansuensis is closest in morphology when it comes to fruit, leaf, and bud traits. Prunus davidiana was considered more closely related to peaches due to pollen morphology similarities. The current cited study, however, holds that Prunus davidiana is more primitive than Prunus cansuensis, making Prunus cansuensis a more likely candidate as the direct ancestor to peaches. Prunus virginensis is related closely to the common peach, but is distinguished from it by long unbranched leaf veins and unique grooves on the peach pit. Isozyme analysis, alongside the common traits, seem to suggest that this species is indistinguishable from the common peach. This concludes part 1 on my series on peaches. Stay tuned for part 2, where we'll talk about cultivar improvement and some cool Frankensteining. See you then, and thank you for watching.